Cellular respiration, chapter 7 is a more difficult chapter. I'll try to give you the high yield points as we go along. So, cellular respiration is going to be the process in which we take glucose, and you can see it being depicted here in the molecular formula as C6H12O6. And we're going to take this glucose and this oxygen that's present, and we are going to break that down into 6 molecules of carbon dioxide and 6 of water. So as you can see here on this picture, that photosynthesis, we were taking sunlight and we were actually creating glucose, right? We were storing energy in the form of glucose. Now this stored energy is actually going to be released through these biochemical reactions that we were previously discussing. So cellular respiration is going to have your book will tell you 4 different phases. I will tell you that there's 3. There's going to be glycolysis, there's going to be citric acid cycle, and then the most important one, the electron transport chain. So you can see here that you have glycolysis. Glycolysis generates pyruvate, and pyruvate is just going to be a fancy word for a substrate. Pyruvate then enters into the citric acid cycle and then goes through a series of reactions and generates acetyl-CoA, as we'll discuss later on. And then it's acetyl-CoA that enters into the citric acid cycle. So this is just kind of a brief overview of cellular respiration. I want you guys to know that within the electron transport chain, we are going to generate the most ATP. Again, within the electron transport chain, we are going to generate the most ATP, 36 to 38 molecules of ATP. So here, again, I'll just kind of walk us through this. We have glucose being a high energy molecule, and it's going to be broken down into carbon dioxide and water, which are low energy molecules. Energy is released that will be used to produce ATP molecules. And we know that the cell carries out cellular respiration to build up ATP. And we know that ATP, through the breaking of the high energy phosphate bonds, is what generates the energy. The most important thing on this slide is knowing that cellular respiration is an aerobic process. And whenever I say aerobic process, I want you to think of requiring oxygen. There is a process called fermentation in which oxygen is not present. Let's say you've had a marathon runner who's gone through a strenuous 20-mile run. When there's not enough oxygen present to feed the muscle, they will undergo something called fermentation. And the byproduct of fermentation is lactate or lactic acid. And this lactate is going to be necessary because it's going to regenerate these coenzymes over here, these cofactors, NADH and NAD+. It's primarily the NAD+, which will be needed to re-enter this glycolytic pathway. And you're asking me, Shay, what is NAD+ and what's NADH? And we'll see this later on. But NAD+ and NADH is something called nicotinamide adenine dinucleotide. And it's just a fancy word. We'll just call it NAD+ and NADH from here on. But it's these cofactors and these coenzymes which accept electrons. And it's the transfer of these electrons through something called a redox reaction. Do you guys remember what a redox reaction is? A redox reaction is just a reaction in which you have either a gaining or losing of electrons, right? We know that redox stands for reduction and oxygenation. And do you remember the mnemonic that I told you how to remember this by? That's right. Oxidation is losing electrons, and reduction is gaining. Exactly. So here, this is a big, lengthy slide. So cellular respiration, like we said previously, was just the breakdown of glucose to form carbon dioxide and water. So it's a process in which we make ATP molecules, right? And we take these ATP molecules, which is stored energy, and we break those phosphate bonds. We can break those phosphate bonds, which releases the energy. So ATP, I want you to know this number right here. I want you to know that in cellular respiration, we generate typically 36 to 38 molecules of ATP. That's mostly the important thing on this slide. There's different pathways in which the glucose molecules can be released. We'll see that later on. And why do we need ATP? Do you guys know? Well, ATP is responsible for many different cellular reactions. So remember the amino acids we talked about previously; those were the building blocks of protein. Well, ATP needs to be present in order to take those monomers and make them polymers. So we can eventually join those polymers together to make peptides, and we know that occurs within translation, like we previously saw in chapters 26 and 27. Um, ATP is also needed with muscle contraction whenever actin binds to myosin, as you'll learn if you're going to take anatomy and physiology. There's an actin myosin cross link bridge that occurs during muscle contraction, and ATP is needed. There's actually a phenomenon known as rigor mortis. Rigor mortis occurs whenever there is lack of ATP, and when would that occur? Well, if you're dead, if there's no biochemical reactions occurring within inside the body, there's no ATP present, which means that actin will not release from myosin. So you have a constant contraction, and the body begins to be stiff. Um, so, as we were previously discussing, these coenzymes NAD and FAD. Again, here, this I'm not going to test you on the structure of them. I just want you to know the name NAD, and I want you to know that it's used in oxidation and reduction reactions. I say here that when a metabolite is oxidized, NAD plus accepts two electrons plus a hydrogen ion, resulting in NADH. It's very hard to imagine this actually occurring within the body. But if you go on to pursue biochem or anything else in the biological sciences, you guys will consistently run across NADH and FADH. NADH stands for nicotinamide adenine dinucleotide, but it's much easier, as you can see here, to just call it NAD plus or NADH. If NAD is not working, which it primarily is, there's only a few reactions in the body that utilizes FAD. Then we use FAD. FAD stands for flavin adenine dinucleotide. And I just want you to recognize the name. I don't necessarily know that I'll test this, but just knowing that it is a coenzyme and it's used in cellular respiration. And I'll point out whenever these are important. So there's phases of cellular respiration. Your book will tell you that there's 4. I will tell you there's 3. Not necessarily a big difference, but I just want you to know that glycolysis is going to be the first step in cellular respiration. Okay? Glycolysis, again, is the first step in cellular respiration. It's always tested, and I'm not sure why. It probably will be tested in my class as well. But glycolysis takes place outside of the cytoplasm. And you've always heard, Shay, but the mitochondria is the powerhouse of the cell. Do you know why it's the powerhouse of the cell? Well, the mitochondria is the powerhouse of the cell because most of the biochemical reactions that produce ATP, and we know that ATP is energy, right? Most of those take place within inside the mitochondria. Because glycolysis takes place outside of the mitochondria, it occurs within the cytoplasm. Oxygen is not present, which makes this an anaerobic process. So again, knowing that glycolysis takes place outside the mitochondria and knowing that it's anaerobic, those are 2 high yield principles. Over here on the right hand side of the screen, we'll see that you have glycolysis, you have glucose entering in here. There's a series of reactions, and you have the production of 2 ATP here. This is called substrate level phosphorylation. We'll cover that probably on the next slide or the slide after. I want you to know the products of glycolysis. Glycolysis is going to yield two molecules of pyruvate and a C3 molecule in order to produce 2 ATP. 2 ATP. That is the amount of energy produced within glycolysis. So as you can see here, overall we typically produce 36 to 38 ATP, and glycolysis is only responsible for a minor portion of this, only two molecules of ATP. So you're asking me, what is pyruvate? So pyruvate is just this chemical structure here. It's essentially just a substrate. So we have glucose over here. Glucose is going to be broken down over here. It's going to be broken down into two molecules of ATP, and over here you can see through this substrate level phosphorylation, we're going to have the exchange of NAD plus to NADH to yield this pyruvate. So just want you to know that it's broken into two molecules of pyruvate and two carbon-3 molecules. After glycolysis, we have something called the preparatory reaction, in which you take that pyruvate molecule. Remember, we just said pyruvate was a substrate, and it's going to be oxidized. So it's going to lose electrons to something called an acetyl group. That might be over your head right now, but again, it's just going to yield something called acetyl-CoA. Moving on, the citric acid cycle is then going to be a series of oxidation reduction reactions that gives off carbon dioxide and produces 1 ATP. So going back to this picture over here, you have glycolysis. Glycolysis is going to, if oxygen is present, we're going to undergo cellular respiration. We're going to produce pyruvate through glycolysis. Pyruvate is a substrate that is going to then enter into the mitochondria, go through this preparatory reaction, and then produce acetyl-CoA. That's what I want you guys to see from that. Acetyl-CoA then enters into the citric acid cycle, right? And there's a series of crazy reactions in here that I had to memorize in my biochemistry class that I will not make you memorize. But I just want you to realize that there is then a transfer of electrons that it goes to the electron transport chain. While we're on this picture, from there, the electron transport chain is then going to do the majority of the work within the mitochondria. And as you can see here, glycolysis is taking place in the cytosol. The citric acid cycle is taking place within inside of the mitochondria. This preparatory reaction is within inside the mitochondria, the electron transport chain in the mitochondria. The citric acid cycle turns twice because the two acetyl-CoA molecules enter the cycle per glucose. Altogether, the citric acid cycle accounts for 2 intermediate ATP molecules per glucose. This is just a picture kind of demonstrating all of the reactions that occurs as pyruvate is turned into acetyl-CoA through that preparatory reaction that we discussed. But I just like to I don't really include that step, but it does occur. Acetyl-CoA comes in, and then acetyl-CoA goes through these series of reactions that we'll just call it magic. There's some magic that occurs, and from there you have the production of ATP. There's a bunch of writing here that you can read. The electron transport chain is a series of membrane-bound carriers that pass electrons from one carrier to another. So after we have this production of ATP, and we have the transfer of electrons within the citric acid cycle, the electrons will then be transported over to this electron transport chain, where you're going to have a bunch of high-energy electrons being delivered to different carriers, and it's through the transfer of these from a high energy carrier to a low energy carrier, which we're going to produce this ATP. So the electrons from one glucose molecule passes down the electron transport chain, contribute to the production of the majority of the ATP between 32 and 34, depending on the type of cell. So I just want you to understand that two molecules of of ATP come from glycolysis. What did we say? Around one ATP comes from the citric acid cycle, and 32 to 34 comes from cellular respiration. Here you have another overview. This is everything coming together, what we've previously discussed. You have glycolysis. We know that glycolysis is going to yield pyruvate. Pyruvate and two C3 molecules, those two acetyl groups, right? It's through that preparatory reaction in which we have the acetyl groups, which are going to combine with coenzyme A to make acetyl-CoA. And acetyl-CoA is what enters into the citric acid cycle. So once you have acetyl-CoA, which is your substrate, that comes into the citric acid cycle, there's then going to be a series of reactions that occur. Again, not really important for our purposes, but we're going to produce 2 ATP. I know the slide said 1 ATP, but it's 1 ATP per acetyl group. And remember, we had 2 acetyl groups, which produce 2 ATP. So here you can see that the electron transport chain, again, all of this taking place within inside the mitochondria, producing 32 ATP. So if I were to ask you a question, which of the following reactions occurs within the cytoplasm? Glycolysis, the citric acid cycle, the electron transport chain, or the preparatory reaction? Which one would you tell me? That's right, glycolysis. Glycolysis takes place within inside the cytoplasm. And you know the other 3 take place within inside the mitochondria. So again, another picture just kind of dictating what I previously just said or depicting. So you have glucose through glycolysis producing pyruvate. You have a preparatory reaction, some magic happens within here to produce that acetyl-CoA entering into the Krebs cycle, also known as the citric acid cycle. And then you have the weird magic that happens in here, producing 2 molecules of ATP. And then we know that we have the majority of our ATP being produced here by the electron transport chain. So again, just getting a little bit more specific. So again, this is going back to discuss the very first phase of cellular respiration. We say here that glycolysis is the initial stage of cellular respiration. We say that it takes place in the cytoplasm. If you don't know that by now, then you've not been listening. It results in the breakdown of 2 pyruvate molecules into a small amount of energy. Do you guys remember how much ATP is produced from this breakdown? That's right, 2 molecules of ATP, as you can see here. This is known as something called substrate level ATP synthesis. So, substrate level, meaning that this occurs by the substrate. This occurs by the transferring of phosphate bonds, the transfer of ADP, and adding another phosphate. And here is an example of substrate level phosphorylation. You can see that there is a transfer of a phosphate from adenine dinucleotide phosphate, right, to adenine triphosphate. Okay. Just want you to be familiar. I want you to associate the word substrate level ATP synthesis with glycolysis. So again, the title of that slide was what? Outside the mitochondria. And we know that it takes place where? In the cytoplasm. Exactly. So again, we said that when oxygen is present, it's glycolysis, cellular respiration is going to occur. But in the absence of oxygen, there's a process called fermentation. And we discussed previously that fermentation is going to produce what? If you guys knew this, I would be very impressed, but that's going to produce lactate. So to correct for if oxygen is limited, pyruvate accumulates in the cell, and intermediates such as NAD plus and FAD cannot be recycled. To correct for this, cells may utilize anaerobic pathways such as fermentation. So we know that fermentation is an anaerobic process. Fermentation is going to yield alcohol and lactate, but primarily I want you to know lactate, right? So you have this marathon runner. This marathon runner has ran 20 plus miles. In order to regenerate the NAD plus that is needed, you're going to produce something called lactate or lactic acid. It's actually what makes the muscle sore because there's no oxygen present. So fermentation consists of glycolysis followed by a reduction of pyruvate and NADH and hydrogen ion. So fermentation, as you can see here, is not very efficient for producing energy. You only get a yield of 2 ATP molecules per glucose. But primarily what it's responsible for, it's this regeneration of NAD plus molecules that so it can re-enter into the citric acid cycle so we can eventually get down to the electron transport chain. So commonly used in muscle cells when our muscles are working vigorously over a short period of time. This results in the buildup of lactic acid, like we previously discussed. So here we talk about the preparatory reaction. We are finally inside of the mitochondria. Um, this preparatory reaction, I don't have really a whole lot to say about it. I just want you to realize that um, it's there. This is what makes acetyl-CoA. So the acetyl groups were inside the mitochondria. We've now gone to the citric acid cycle, which is actually located within the mitochondrial matrix. I'm not sure if you guys remember discussing that within the, I believe it was chapters 3 or 4 when we were discussing the organelles. We were discussing the composition of the mitochondria, and we discussed something called the mitochondrial matrix. This is actually the location within inside the mitochondria where um, the citric acid cycle will take place. And we know the citric acid cycle is where all the magic happens. It's actually where a lot of these reactions here that we previously saw. Um, so all of this is taking place within inside that little area called the mitochondrial matrix. And this is going to be responsible for the production of NADH, CO2, as you can see here, a molecule of FADH2. So here, we produce one ATP, but we know that the entire cycle must turn twice per glucose molecule, so we actually produce 2 ATP. And then the final stage of breakdown involves the electron transport chain. And again, this is our big boy. This is the one that's going to produce between 32 to 34 molecules of ATP. And it's important to know where the electron transport chain is located. It's located within the cristae of the mitochondria. That's always tested. Commonly tested questions. Glycolysis takes place where? In the cytoplasm. What reactions take place within the mitochondria? Well, we know the citric acid cycle takes place there and the electron transport chain both take place there. Then they can get more specific and they can say, well, where does the electron transport chain? Which portion of the mitochondria? It's actually within the cristae, whichever way you want to say it. And then they can say, well, where does the citric acid cycle takes place? And we know that is within the mitochondrial matrix. Um, so again, here we see the electrons received from NADH and FADH2, which we previously saw in the citric acid cycle. That's passed down a chain of electron carriers, which we'll see later on, finally received by oxygen. Remember, we said that cellular respiration was an aerobic process. So the final electron acceptor is oxygen. Again, the final electron acceptor is oxygen. And we know that these electrons from NADH and FADH2 are passed on through a series of redox reactions. You know that redox reactions are just transferring of electrons. We know that when molecules are oxidized, they lose electrons. When molecules are reduced, they gain electrons. Here you can see the complexes. There's 4 complexes located here on the right side of the screen. You have complex 1, complex 2, complex 3, complex 4, and there's something called cytochrome C here. If you watch the evolution lecture, then you'll see I began discussing about the composition of cytochrome C in relations to humans and primates, and I talked about the very specific 97% specificity between cytochrome C located within humans and located within primates, and this was the structure I was talking about, and that was located within the mitochondria. Not to get off topic, just wanted to just kind of clarify something I'd previously said. Not to labor on much longer, but there's something called chemiosmosis. And chemiosmosis is simply when hydrogen ions fall down the groups, they go through these ATP synthase complexes. Energy is released and used to form ATP molecules from ADP and phosphate. So I want you to associate two words, right? I want you to associate substrate level phosphorylation with glycolysis. And then I want you to associate chemiosmosis with the electron transport chain. When someone says glycolysis, you should automatically say substrate level phosphorylation. When someone says electron transport chain, you should automatically say chemiosmosis. So this is how energy is created. It's created through the passage of these hydrogen ions, and it falls down these groups here. And it's just the transfer of these molecules. So we see here that ATP production, right? We see that there's variable numbers. Again, I'll make it very apparent on the exam, but glycolysis produces 2 ATP molecules. The citric acid cycle produces 2 ATP molecules. We know that it produces 1 ATP per glucose molecule, but because there's 2 glucose molecules, it turns twice and it makes 2 ATP. And the rest are produced by the electron transport chain. So the yield's actually closer to 30 to 32. Thank you.